Hello everyone, today I'm going to present a deep learning architecture capable of solving multiple tasks for quantitative microscopy image analysis by learning only a simple classification task. A quantitative analysis relies on the extraction of quantitative parameters such as the number of cells or the average area of a cell. Commonly, such parameters are extracted manually from the image using different annotations. For example, precise contouring of cells or marker annotations. Unfortunately, this is a long and tedious process which must be repeated on multiple images. To reduce the burden of manual annotations, deep learning models were introduced to learn a mapping between the input image and the desired annotations. Hence, it is expected that at the inference step, the model should be able to extract similar characteristics from the image given a high number of training examples. Unfortunately, a precisely labeled dataset is not always available or may be difficult to obtain. Hence, we set out to develop a learning model capable of producing detailed feature maps from training using only image-level annotations, which we termed MICRA-Net. In other words, MICRA-Net aims at predicting the presence of a structure within the field of view. At inference, the presence of specific structures within the image is predicted, after which the class-specific gradient class activation maps are extracted from the architecture at each layers. We then merge the low and high level features using a PCA to generate a single detailed feature map that we can then manipulate. We challenge MICRA-Net on a wide variety of tasks, but today I'm going to focus on the semantic instance segmentation task. We use the publicly available cell tracking challenge dataset. We aimed at predicting both the semantic presence of the objects within the field of view and the possible contacts between cells. Hence, we can simply remove the contact between cells to generate an instance semantic segmentation map. Here are some examples of predicted segmentation. Remember that MICRA-Net was trained solely using image-level annotations. Before comparing MICRA-Net with fully and weakly supervised state-of-the-art methods, we wondered about the level of supervision that can be obtained from annotators using a user study. We asked different annotators to precisely annotate the objects within the images. To approximate the level of supervision, we altered all objects in this segmentation mask of the ground truth using dilation and erosion with a size randomly sampled from a normal distribution. We then compared the intersection over union of the annotators and the altered version of the dataset with the non-altered ground truth annotations. We were able to show a supervision level of alt 5 for the annotators. We compared MICRA-Net with fully and weakly supervised state-of-the-art methods. The weak supervision consisted in the altered version of the training dataset and bounding boxes. We used the area under the F1 score as a function of minimal IOU between associated objects. We compared MICRA-Net with the baseline. Here, bluer is better. Only a unit trained with minimal or full supervision obtains a significantly higher performance. Interestingly, MICRA-Net is superior or equal to state-of-the-art methods trained with a supervision of alt 5. The level of supervision of the precisely annotated dataset from the user study. Hence, when the level of supervision of the training dataset cannot be guaranteed, MICRA-Net should be considered, since it can achieve a similar performance with easier to obtain annotations. I thank the members of the lab and the funding agencies, and thank you for your attention.